네, 안녕하세요. 저는 서머피셔 바이오 프로덕션 그룹에서 마케팅과 비즈니스
Good
디벨롭먼트를
afternoon. I
담당하고
am
있는
from Thermo
김현주라고
Fisher.
합니다.
My name is Im
우선
Yeon-mi.
오늘 귀중한 시간 내주셔서
Thank
참석해
you very
주셔서 정말
much
깊은
for
감사를
sharing your
말씀드립니다.
valuable time to join us
오늘 저희
today.
서머피셔에서는 멀티오믹스와
Today, in this
바이오인포메틱스를
session,
활용한
we would like to talk about
셀컬처 미디어
the cell
디자인에
culture
대해
media
글로벌
design using
연장부에서
multi-omics and
온라인으로 잠시 후
bioinformatics.
발표를 진행을 해주실
So
예정입니다.
our global speaker will give us a
올해로
presentation
창립
shortly.
60주년을 맞은 깁코는
GIPCO
셀컬처
celebrates the
미디어와
60th-year
서플리멘트
anniversary
등의
this
세포
year,
배양 원료 물질뿐
and
아니라
not
미디어
just
디벨롭먼트
for the
서비스,
cell
셀라인
culture
디벨롭먼트 서비스
materials, but also
등의
media
다양한
design
솔루션을
service and cell
바이오랩
line
운영 및
development services have been
생산을 위해서
developed
공급을
and
하고 있습니다.
provided by GIPCO. For bio
깁코의
research,
미디어 디벨롭먼트 서비스는
especially the
고객사의
GIPCO's media
셀라인의 최적
development
컨디션 운영
service
기술을
is
이후
to
생산
develop
단계까지
optimized
원활하게
media for
스케일업이
customers,
가능하도록
and also
토탈
even for
솔루션을
scale
제공드리고
up, it
있습니다.
provides a very comprehensive solution.
오늘 소개해드릴
So
멀티오믹스는
today we would like to talk about the
프로테오믹스와
multi-omics, which
메타볼로믹스를
is
기반으로 한
based on the
셀컬처
proteomics
미디어를
and
디자인하는
metabolomics.
최신
And this
기술이며
is a state-of-the-art technology
발표를
for
진행해
cell
주실
cultural design and
신진투
development.
박사님에 대해
And
제가 간략히
I would like to
소개를
briefly
드리고
introduce to
세션을
you the
시작하도록
speaker,
하겠습니다.
Dr. Chung Jen-tu.
청진투 박사님은
Dr. Chung Jen-tu
상하이
is
인스티튜트 오브 바이오케미스트리를
working in Shanghai, and he graduated
현재
from the Institute
서머피셔
of
사이언스
Science
및
in Shanghai,
바이오
China,
프로덕션 그룹의
and currently
글로벌
he is a
R&D
senior
업무를
staff
담당하고
scientist
계십니다.
in bioproduction group of Thermo Fisher
주로
Scientific.
멀티오믹스와
He's mainly in
바이오인포메틱스를
charge of the
플랫폼을
multi-omics
적용하여
and
세포 배양 매체를
bioinformatics and
최적화하는
apply
업무를
them to
담당하고
platform
계시며
to optimize
멀티오믹스를
the cell
활용한
culture, and
깁코의
also
다양한
utilizing
세포
the
배양 매체를
multi-omics. He
성공적으로
develops
개발하고
the
출시하신
various
분입니다.
media for GIPCO.
오늘 강의를 들으신 후 질문이 있으신 경우
After his
세션
presentation,
마지막
if you do
질의응답
have any
시간에
questions,
부탁을
please raise them during the Q&A
드리겠습니다.
session, which is at the end of the
아쉽게도 시차
presentation
때문에
session.
라이브로
Because of the
Q&A를
time
직접
difference,
참석하는
Mr. Chung Jen-tu could not be joining us on a real-time basis, and I would like to ask for your understanding. But we have Mr. Kim Hyojin from Asia Pacific Thermo Fisher Scientific, so he can address your question, or later we will get back to you through the email. Now we would like to invite Chung Jen-tu for his presentation.
Good afternoon. My name is Chen Jiantu, and I'm a senior staff scientist in the bioproduction group at Thermo Fisher Scientific, Grand Island, USA. Our bioproduction group grew out of what is called GIPCO, the Grand Island Biological Company. This year is actually GIPCO's 60-year brand anniversary. In today's presentation, I wanted to talk about multi-omics and bioinformatics in cell culture media design. I have divided my presentation into four main parts. First, I will give a brief overview of analytical methods for media development. Next, I will introduce the concept, benefits, and the workflow of multi-omics in media design. Then we will take a closer look at these technical details in a case study. At the end of the presentation, a brief summary will be provided. Okay, let's start talking about the analytical methods for media development. Titration of individual components is the traditional strategy used for cell culture media development. It involves conducting a series of experiments to determine the dose response of a cell line by adding each component at different concentrations while keeping the others constant, and try to find the effective range for each component. As you can see, this method has been successfully applied to develop first-generation cell culture media, such as F12 and MCDB301. I will say this method works great. The problem is that it is time-consuming and does not consider the synergistic effects of components. Then, new technologies such as design of experiments, DOE, and statistical analysis have been applied to media development, allowing scientists to evaluate multiple components or media mixture at once and identify their interactions. This statistical DOE approach usually relies on high-throughput automated cell culture bioreactor system. Through statistical evaluation of properties such as cell growth and titers, the optimal media mixture and the optimal concentration of various components can be identified. Further optimization can be achieved when performing spent media analysis. Scientists can calculate component consumption rates based on spent media analysis and assess the impact on key metabolic pathways such as glycolysis and the TCA cycle. Using statistical algorithms, metabolically balanced median and feeds can be generated. I will say that DOE, small-scale bioreactor, and spent media analysis are the three essential elements of current median development. This is actually our traditional way of developing cell culture medium at Grand Island. Recently, we have incorporated multi-omics into our cell culture medium development pipeline, which allow us to monitor more than 8,000 molecules and get better insights into nutritional requirements for cell growth and protein production of CHO cells. The main idea here is to use multi-omics to identify the bottlenecks that might be missed by traditional approach. Next, I will introduce more details about multi-omics. In this part, I will introduce what is multi-omics, why we should use multi-omics, and how to utilize multi-omics in medium design. So, what is multi-omics? From genotype to phenotype, there are many different omics such as genomics, epigenomics, transcriptomics, proteomics, and metabolomics, or maybe lipidomics. Multi-omics aims to combine and integrate two or more omics datasets to investigate biological pathways in a more comprehensive manner. For an example, to investigate what's under the puzzle, we can use non-targeted omics such as transcriptomics, proteomics, metabolomics to reveal more and more of its contents. Then we can transfer to targeted multi-omics analysis, which can be designed to only monitor the secret smile of Mona Lisa to improve sensitivity and, most importantly, save time. Currently, we are in the discovery stage and using non-targeted omics approach. After we identify more key pathways or networks in CHO cells, we will move to the verification stage using targeted omics analysis. Talking about the benefits. As I mentioned in the previous slide, the main benefit is that multi-omics can identify more bottlenecks than the traditional workflow. That's because the traditional workflow, which is mainly based on DOE and spent media analysis, it only monitors less than 50 molecules and involves multiple iterations to achieve an optimized media, while multi-omics can monitor around 8,000 molecules, which allow us to identify specific pathways and tighter influences, leading to a more directed DOE design to optimize media and feeds. Another benefit of multi-omics is that traditional workflow is usually only to rebalance the nutrients, while multi-omics enables us to launch new components. So how to utilize multi-omics in medium design? First, we need to establish a metabolic baseline, which includes adaptation to a panel of medium with high diversity. Cell culture in small-scale bioreactor and multi-omic sample collection. Then there is multi-omics analysis, including sample preparation, HPLC mass spec analysis, and multi-omics integration and interpretation. In the omics-driven DOE step, key pathways will be selected. Inhibitors and activators for specific pathway will be validated in small-scale bioreactor. Then the final medium formulation and QC of protein production will be provided. Okay, next let's talk about more details in the case study. This is an external service project. The goal is to develop a chemically defined medium to increase titer without affecting product quality. The background is like this. The cell line is Dg44 CHO cell line, and the traditional workflow didn't work well for this clone. And meanwhile, they wanted to keep the original cell culture process. The target is very clear: to develop cell culture medium and feed that allow a threefold increase in titer. In next few slides, I will go into more details on how to utilize multi-omics. For small-scale cell culture, we usually will choose M15, which allow us to evaluate 48 conditions at a time. Traditional fed batch cell culture needs 14 days, so if we collect samples on every day, there would be too many samples. 48 times 14, there are about 700 samples. So considering the feasibility, we reduce the time points to 3, which will cover 3 different stages of cell culture, including early stage, log phase cell growth stage, and the stationary stage. We will collect cells and spend a medium, and would like to perform cellular proteomics, cellular metabolomics, and spend medium metabolomics analysis, 3 omics here. After sample preparation, we will perform proteomics runs on our Orbitrap Fusion Lumas and our metabolomics run on our Orbitrap IDX, and then followed by data analysis and the multi-omics integration, which will lead to the final omics guided DOE design. For metabolite identification, we use Orbitrap IDX tri-breed mass spec, which contains 3 mass analyzers: quadruple, linear untrap, and Orbitrap. So when we use Orbitrap as the detector, high accurate MS1 and MS2 could be achieved. For the database search and quantification, we use Thermo Fisher's Compound Discoverer software. Compound Discoverer has integrated MZ Cloud, which will search against the high-resolution MS2 database, and then identify more reliable endogenous metabolites. CD software also uses the node-based workflow, which makes the database search and statistical analysis faster and easier. For the relative quantification, we use the MS1-based quantification method, which means we compare the mean peak area across different groups. The example here is for L-glutamate, as shown in the red box. It shows a high match of MS2 spectra with the MS2 database, and a significant difference between these 2 groups, orange one and the blue one, can be observed. For our proteomics platform, we use Thermo Fisher Orbitrap Fusion Lumas and Ultimate 3000 HPLC. We have optimized the whole system from head to toe. We have compared different trap columns, analytical columns, and ESI emitters. We also optimized HPLC and mass spec parameters. For the MS2 acquisition mode, we choose HCDOT. That means high energy collision dissociation was used for precursor ion collision and OV-trap was used for detection. The benefit of this mode is that higher protein ID and better quantification could be achieved. Another main optimization I wanted to mention here is the use of larger ID trap columns, which allow us to analyze more than 100 biological samples with high reproducibility. After the installation of FAMES, by now we can identify more than 6,000 proteins from a single run, and more than 7,000 proteins from a whole dataset. For protein quantification, we also use the MS1-based quantification method. Spatial count and MS2-TIC are MS2-based methods. Isolate-based approach here is a MS2-based method. As you can see, MS1-based method provides higher reproducibility, especially for low abundance proteins. For the platform stability test, we have designed an experiment to run 100 times with the same sample, 5 peaks of different intensities from E5 to E10, and different retention times were selected. As you can see, retention time and the peak areas are reproducible across 100 runs. For retention times, the coefficient variations, so-called CV, were below 1% for all the 5 peaks. For protein intensity, after 100 runs, the protein intensity only dropped 20% using our system, while without optimization, the intensity will easily drop more than 50%. After HPLC spec analysis, we will do data analysis. For the fed batch cell culture, we usually select 3 time points, like day 5, day 7, day 10, as I mentioned in the previous slide. Then we do principal component analysis. We can see different days and different conditions were well separated with the first 2 principal components. This usually means that there are significant differences between these groups. Cluster and heat map analysis can first tell the ordered protein and the metabolites. Then we perform an ingenuity pathway analysis to integrate different omics. The changed pathways or networks between conditions with high titer and low titer or high VCD and low VCD will be identified. And then we use those information to guide our next DOE design. In addition to utilizing bioinformatics tools for mass spec analysis, Multi-Omics data integration and interpretation, we also use Multi-Omics tools to select the appropriate panel median for specific cell line. Great diversity usually gives us great possibilities. So bioinformatics analysis is widely used at every step of our Multi-Omics guided median design. In this slide, I just wanted to show high quality and reasonable results from metabolomics and proteomics. For those small molecules, such as those low abundance vitamins, the intensity patterns from the positive mode and the negative mode are very consistent. Here, the variations are from biological replicates, not the technical replicates. For those quantified proteins, such as glutamine synthetase, which help to generate glutamine from ammonia and glutamate. If this enzyme level is low, the consuming of glutamine usually will be higher, right? The cell needs more glutamine, which also means we need to add more glutamine for this cell line during cell culture, just like for SP20 cell. For the pathway analysis, we use ingenuity pathway analysis to identify key pathways and select pathways that are easy to manipulate. For example, we found that the mTOR pathway was positively correlated with titer. More more than 30 proteins and metabolites suggested that the mTOR pathway was activated or more active in conditions with higher titer. Here it shows the selected 6 key molecules involved in the mTOR pathway. Then we will design different components or a group of components based on this information and try to manipulate those pathways with those design factors. After we identify these factors to manipulate the selected pathways, we will start the first AMBER experiment to screen out the effective components. As we all know that different cell clones respond differently when we treated with the same components at the same condition. So for each pathway, we will design several different factors and want to select out the effective ones. The figure shows the common features such as BCD cell viability and titer from the top 5 conditions. As you can see, these 5 factors have different growth performance, indicating that they are likely to have a synergistic effect. So these 5 factors were selected for a next AMBER experiment. 2-level fractional factorial DOE design was used to evaluate the synergistic effects of these 5 factors. The top 5 conditions for each feature were shown here. Since the results from these DOE conditions already met our goals, so we stopped further optimization, then move on to evaluating the QC of the antibody and wanted to identify the basal median and feeds that provide the same quality as customer control. There are so many signaling and metabolic pathways that affect the same downstream effects, such as cell growth, right? Our Multi-Omics approach allows us to identify specific pathways for specific clones, which can then be manipulated further. In this project, we achieved a 3-fold increase in titer after the DOE verification step, and our customers are very happy with it and tell us that the titer improvement is impressive and competitive enough against other DG44 improvement options. For the deliverables, first, we will introduce our Multi-Omics process and share the study plan. Second, high-quality data will be generated through strict quality control. Then we will use statistical analysis to dig deeper into bioreactor GPA results and the Multi-Omics data. The top 10, 20 key pathways for your clone will be explained further. Inhibitors and activators will be designed to manipulate these pathways. After validation in the same cell culture system, omics optimized medium, feed, and the cell culture process will be provided. In addition to Multi-Omics, here I would like to remind that our Giveco services team offers our clients an excellent portfolio of medium solutions, no matter where they are in the process. From cell line development to final medium manufacturing, the spec-based omics technology can help at nearly every step, such as comparison of different clones, transfection efficiency troubleshooting, quality control for all the schools or catalog media. Today, our Multi-Omics approach is mainly used in media and feed development. For external customers, we will provide a quick and efficient way to narrow down the ideal media, such as catalog or panel media. If needed, Multi-Omics-based optimization will be followed further to meet specific goals. To wrap up, there are several take-home messages. First, current multi-omics technologies allow you to monitor about 8,000 molecules and help you identify and resolve multiple bottlenecks at once, unlike traditional methods that only monitor 30 to 50 metabolites. Second, we have successfully applied multi-omics to more than 10 projects in cell culture medium optimization and achieved promising results. In 2 of them, we used the multi-omics and the traditional approach in parallel. As expected, multi-omics always provides us better results. Third, bioinformatics analysis is not only important in multi-omics analysis, but also in data interpretation. Our next step is to incorporate multi-omics into our machine learning models to enable more accurate predictions and decisions. Please let us know if you have any questions. Our colleagues will help us to collect your questions. We will get back to you as soon as possible. Thank you for your time.
Thank you very much.
혹시 질문 있으신
Do you have some questions? If you have questions, please raise your hand and our staff will give you the answer.
질문 부탁드립니다.
우선 강의 정말 좋은 정보 주셔서 감사하고 잘
So
들었습니다.
thank you very much
질문이 하나
for the
있는데
presentation. I have one
대지
question.
개발 방식에
As
있어서
for the media
기존
development,
방식과
so
multi-omics를
multi-omics
활용하는
and the
방식의
traditional
가장 큰
methods,
차이점이
what is the biggest difference between the two?
네. 켄진 박사님께서 설명을
As
해주시는데요.
explained by
실제
Dr.
기존의
Chen
배지
Jun,
개발 서비스는
so the
그런
traditional media development
이런
service,
멀티오믹스 어프로치가 아니라
it's not based on
실제
multi-omics.
실제 배지를 개발하고 그 DOE를 통해서
Rather, it is through DOE after media development. So after several rounds of iteration, its focus is more on rebalancing.
실제로 모니터링할 수 있는 몰큐리 한 40.
And also the actual molecules that can be monitored is only 40 to 50.
기존의 회사들은 그런 방식을 통해서
So,
잘 개발해 왔지만
Yes, companies have succeeded, but the Thermo Fisher developed a new multi-omics approach. And through the metabolomics and proteomics, we can actually monitor 7,000 to 8,000 molecules all at once. And by doing so, we can produce the big data. And then through the data analysis tool that we have, we analyze the key pathways for each cell. And then we finally develop the optimized media for each cell. So we do have a lot of biologic companies, and they make huge investment to develop biologic products. However, developing the optimized media can actually make the best use of the potential of the cells. And in this sense, I think multi-omics-based media development service, we can identify and develop the key pathways of the cells, and also what activators and inhibitors could determine the productivity. We can identify that as well, and therefore we can effectively develop media using multi-omics.
궁금한 게요. 그러니까 배지 요소들이 서로 유기적으로 다 연결이 되어 있는데 저런
So,
멀티오믹 데이터를 도출한다고 했을 때 어떻게
When
조성을
producing multi-omics data,
조합하는지가 그거에 따른 대응이
how
굉장히
do you
어려운
combine
것
the
같거든요.
components
네.
together?
관련해서 어떤 레이셔널이 있는지 설명해
Do you have any rationale for that?
일반적으로 저희가 이제 제일 중요한
Generally speaking, so this is to analyze some key pathways, and for each cell, the required nutritional value is all different. So what we are doing for analysis is that in the past we only used the spent media analysis, but we look at intracellular and extracellular environments together. And then during the cell culture process, we see how these metabolites are changed. So this is the major thing for analysis. And also we have huge amount of data regarding that. So we refer to our own proprietary database to identify which pathway and which metabolites and which deterrents should be utilized to maximize the performance of the cells. And for this, if you want to have some detailed information, after this session, please contact us. Then we will link you with the R&D team. So maybe we can have a video conference to give you more specific information. Thank you very much for a lot of questions. Because of time constraint, we have to conclude the session here. And the Thermo Fisher's booth is located on the first floor. So if you do have any further questions, please visit our booth. Thank you.